Welcome back to this next section of Introduction to Neuroscience, where we're going to continue on our journey to understand how it is that the cells of your brain, the neurons in your brain, are able to process and represent information that allows you to do all the things that you do in your normal lives, including watch this video. So in the last sections of videos, what we did was derive a set of passive as well as active properties of neurons as electrical cells. And at the end of it, we made the approximation of the cell as a cylindrical body, and that allowed us to have an action potential, this all or nothing self-reinforcing wave of electroactivity that is able to very efficiently transmit down the length of an axon, this part of the cell here, without loss of information. Now, this was really great, super cool, right? And we've already learned about different pathologies of the process of generating this action potential that's been relevant in different types of diseases of human relevance, as well as different targets of toxins. However, this actually leaves us with even more questions. So it's really cool that we have a cell that's not just a cylinder, but actually has more shape. So we have an action potential, but where are action potentials generated, right? In the last section of the videos, we basically said, assume there starts an action potential, it can keep going. But how do we start one? At the same time, there's the question of what happens when the action potential gets to the end of the cell, right? According to the Neuron doctrine, cells are distinct cellular structures and they are not connected to each other in some kind of weird continuum. And so surely there must be an end of the axon, the end of action potential. So what happens at the end of the action potential when the action potential actually gets to the end of the line, okay? When there's a train station down there, what happens? Everyone has to get off. So the short answer is that the cell, this neuron, is going to connect to another neuron in a set of structures that is known as the synapse. And so we're going to be talking about synapses, but then it begs the question, are all synapses the same? Are there different types of synapses? We'll learn about that. And we are going to be asking about how can a synapse change? Because after all, the synapse is a fundamental unit of communication between one cell and another cell. And so being able to change the strength and dynamics of the synapse is one of the reasons that your brain is actually able to change and learn in time, so that you're not the same person that you were a year ago. And you have new experiences and new things that you've learned, and you can grow throughout your lifetime. So if you are ready to come on this trip with me, here is the plan. Here's our itinerary for the next couple of videos. So in this video, we're first going to start with just the basics, but by looking at neurons and the anatomy of a neuron and a synapse, so that we know the parts of the neuron that we were talking about. Next, we're going to be talking about neurotransmission at the synapse, what happens when the action potential gets down the end of the line, and the synapse that has to get to the next cell. We're going to be talking about different types of synapses, as well as different types of receptors that happen to receive this information on the next cell over. So these are broadly called ionotropic and metabotropic receptors. Those are the large classes of receptors we'll be talking about. So there's a video on exactly what those are and the different types that are relevant for your brain. All of that leads us to a couple of tangential topics that are of interest to lots of people. This includes topics like recreational drugs in the brain. It turns out that a lot of recreational drugs in the brain target parts of synaptic transmission. And we're also going to be talking about drug addiction and learning. So that's the plan. We're going to go back and start with the cell. All right, here's a neuron. And what you'll notice here is that the neuron has a lot of different processes and different compartments. Rather than being a single compartment that is well approximated by a sphere, or even with a sphere that has a couple of little dendrites and little things coming off of it, it has a lot of different complicated structures. So generically speaking, a mammalian neuron that you might start looking at in an intro to neuroscience class, like this one is, has the following components. There is the cell body, which is this blobular thing in the middle here. Neurons tend to be relatively large cells because they're metabolically very active. So the cell body tends to be pretty big. They're kind of on the big side as eukaryotic cells go. And that is where the nucleus is going to live, and that's where the nucleus is going to stay. Now, generically, what happens is that there is a set of processes coming off of one end of the cell body. The cell body is also equivalently sometimes called the soma. So when I say cell body or soma, they really mean exactly the same thing. One side of the cell body has a set of processes coming out. These are branches that we call dendrites. And those tend to be the inputs of the cell. We'll talk a little bit more about exactly what that means in a couple of minutes. And then the other side of the cell, we have a different process coming out that is called the axon, which are the outputs of the cell. This is where it's going to go and talk to other cells. Now, these cellular compartments are not only anatomically distinct, they look different. In most cases, they are also physiologically distinct. They express specific proteins that support their relative function. We talked about myelin sheath and myelination in a previous video, and so myelin sheaths are one of the ways that you can wrap some axons, not every axon, some axons are wrapped in myelin sheaths so that they are able to conduct action potentials even faster in cases where you might be having to communicate in the distal part of your body all the way down towards the middle. So those are the basic parts of a cell, but I wanted to show you a couple more real examples of what cells actually look like in your body, because that was kind of a cartoon, right? So cartoons are really great. We like looking at them, but let's look at a couple of real cells for a little bit. So here is a pyramidal neuron of the cerebral cortex. These are the major excitatory cells in your cerebral cortex, and what they have is a cell body along with a bunch of dendrites. Okay. What's pointed out here is that these dendrites can actually be quite large. The arborization can expand quite a bit far from the cell body. And so sometimes we talk about apical dendrites versus basal dendrites because these different dendrites, because of their location and relative geometry to each other, also has computational implications. That's something that we'll talk about in a following lecture. And then there's also an axon. So you'll see that in this particular case, it's a variety of different processes that are the output of this particular pyramidal neuron. There are some proximal axons where it might make synapses with close by cells. And sometimes you can have a very long axon that goes to lots and lots of faraway places in distant lands on other parts of your brain. So that's a pyramidal neuron, and here is a neuron that you might find in your spine. So this is a spinal motor neuron of the kind that innervates the muscles in your limbs. So the kind that might be innervating the muscles in my arm so that I can flex and move my arm in this particular way. So you can see here that colored in the way that I've colored it with dendrites are orange and axons are blue. It's really easy to tell them apart that there's dendrites and their axons. But if you're looking at it just under the microscope where you have the cells stained but you don't have this clear distinction, sometimes in these spinal motor neurons it's actually difficult to tell which of the processes coming off of the cell are dendrites and which of them are axons. So that could be really complicated. You can see in this example cell here there's actually lots of processes coming off of the cell body. Most of them are dendrites and only one of them is an axon. So that's one thing that we see sometimes. Here's another type of neuron that we see. This one happens to be a sensory neuron that also lives in your spine. It's in the dorsal root ganglion cell of the spine. And so in here, there's a funky set of geometry here where the cell body lives in your spine. One end of the axon goes towards your central nervous system, goes towards your brain. The other end of the axon goes towards the periphery. And then it terminates in a set of nerve endings that takes in sensory information from distal parts of your body, like the tips of your fingertips or maybe the tips of your toes and stuff like that. This is the neuron that is the example of the longest cell in your body. Okay? So for comparison, what we're looking at here is there is one particular set of neurons, which are sensory neurons in the dorsal root of the ganglion of the spine, that is responsible for innervating your tippy toes. And so its cell body is going to live in the last vertebrae in the bottom of your spine. One end of this axon goes all the way to your toe, and the other end of the axon goes to your brainstem. So this cell is practically as long as you are. Now, from what we understand about vertebrate neuroanatomy, not only do humans have this cell, it's quite likely that even extinct vertebrates also had the same cell. And so even though this cell has not been preserved because we can't really do live paleoneuroscience, even though I desperately want to, it's quite likely that the cell in one of these really large extinct dinosaurs, here we're looking at the Argentosaurus, is the longest cell that has ever existed. We have no direct evidence that the cell did exist, but we have fossil evidence that these dinosaurs exist, and they were in fact this large. And so if their neuroanatomy looks like every other neuroanatomy that we know in other vertebrates, presumably it had a cell of this kind also. So that's kind of cool. Okay. So those are some vertebrate cells. I wanted to show you at least one example of a neuron from an invertebrate. In this particular case, this is an invertebrate neuron in the fruit fly ventral nerve cord, which its computational equivalent of a spine. Instead of having a spine in the back of their body, flies are examples of invertebrates that their nervous system is actually in the belly and their digestive tract is in the back. And so this is the ventral nerve cord. And this particular motor neuron, you'll see that it looks a little bit different than the motor neuron that we had in the mammalian spinal system. It has a cell body and it has only one process coming out of it. There's some sub-branches of that one branch, those are the dendrites, and then the branch keeps going, that's the axon, where it terminates in the muscle and commands the fly's legs or wings to move, just like the way it does in us. So there's a lot of commonalities and also some subtle differences. And what we're going to do now is think about the flow of information in the generic sense. We're going back to the cartoon version of the neuron. And what we're going to see here is that the flow of information, generally speaking, there are exceptions, generally speaking, goes from the very ends of the dendrites to the cell body down the axon. That's the flow of information. And this is the flow of information the majority of the time. So let's talk about this case first, and then we can talk about exceptions later. So the majority of time what happens is that this cell is going to be receiving input information from the rest of the nervous system. So for example, at one tip of this dendrite, you might be receiving a positive input from another cell, causing some kind of depolarization. So we're seeing here a small depolarization where the membrane voltage is going more positive, so it's depolarization. From this particular branch of the dendrite, maybe you have something that is what's called an inhibitory synapse, where you're receiving a hyperpolarization and the membrane voltage at this location is actually going more negative. And so on and so forth. So you can have lots and lots of these neurons that are having the effect of having summing inputs into the dendritic tree of the cell. Now, as we learned about in an earlier video, the passive and active properties of the neuron tell us what happens when you have either depolarizations or hyperpolarizations from the resting memory potential. What's going to happen is that the time course of the depolarization or the hyperpolarization is going to be exponential, following Ohm's law, a derivation of Ohm's law, where you solve that differential equation over there. You can review that video for details if that's something you would like to know more about. And because these depolarizations are then in the membrane of the cell, they're local, they're going to be propagating down the branches of the dendrite following the Cable's equation, where they're going to be propagating outwards, and when they meet each other, they're going to add, right? So two positives are going to become more positive, and a positive and negative might cancel each other out to some extent. Now, when all of that goes all the way down and propagates down all of the various axons, I'm drawing 3 here, but in reality, there's thousands of these things for every cell. It's going to go down, hit the cell body, okay? And there's a special spot at the very base of the axon where the axon initially comes out of the cell body, comes out of the soma. It's a special spot called the axon hillock. And the axon hillock is special because this is the point at which an axon either generates an action potential or don't. This is an all or nothing event. Now, you can kind of imagine that if you got a small depolarizing potential of some kind of in a far away distant part of the dendrite, by the time it's made its way, followed the Cable equations all the way down to the cell body, it's pretty darn small, right? So if it's really small and it doesn't pass some kind of threshold, then an action potential will not be triggered. There'll be no action potential. But if you had a really big depolarization and it's kind of close to the cell body, then maybe the version of that depolarization that propagated all the way down to the axon hillock will be large enough, maybe when combined with a couple of other ones that also happen at the same time, to actually pass some kind of threshold to trigger the voltage-gated sodium channels at the axon hillock, thus generating the feedback loops to initiate an action potential. Once an action potential was generated, then it maintains its shape and it passes as a wave all the way down the axon and reaches other parts of wherever the cell is trying to talk to. Okay, so that's cool. And if you want to review the details of exactly how an action potential is generated as well as regenerated as a self-reinforcing set of feedback loops, please see the video on the action potential. So, and to summarize, we have inputs coming through the dendrites, where you have passive and active properties of neurons that apply here, but we don't have voltage-gated sodium-potassium channels. And so generically speaking, even though there's exceptions, there are no action potentials in dendrites. Action potentials are generated at the base of the cell body called the axon hillock. This is where there's a large concentration of voltage-gated sodium channels. And so we can get, once you pass a threshold, a self-reinforcing wave of action potential, which then propagates all the way down the axon. That's the generic version of what happens. Like I said, exceptions do exist. And as we learned about in the action potential video, these action potentials, because the voltage-gated sodium channels are inactivating, cannot propagate backwards. So once it's generated at the axon hillock, it's going to go forwards, and it's unable to travel backwards, upwards towards the cell body. This is a generic version of what happens. Okay, we're going to be looking at this diagram and this idea of information flow a couple of different times throughout the next couple of throughout the next couple of videos, and we're going to be building on this intuition to try to make the equivalency of what happens between biology and biochemistry and protein chemistry, and also the mathematical analogies between what the neuron is actually doing when it's computing in this particular way. So that is what happens. I've walked you through the process of the cell receiving inputs, deciding whether or not to generate an action potential, and then the action potential propagating all the way down to the end of the axon terminal. So what happens at the end? All right, I've already given you a preview here. What happens at the end is a special set of structures in your brain called synapses. Now, recall that the human brain has approximately 86 billion neurons and is about the size of two of your fists put together. All right. Now, on average, at least in the human brain, each neuron makes about 1,000 to 10,000 synapses. Now, if you just do that multiplication in your head, that is a heck of a lot of synapses. Instead of talking about all of them, we're just going to focus on a couple of different ones. And in particular, I'm going to show you an electron micrograph of just one synapse right now. So here is what a synapse looks like if you zoom way in and you look at the structure of it. So what we're seeing here is a place where two cells are so close to each other that they've actually met. They're touching because they're associated with basically protein Velcro. That's making sure that they stay together. However, they're not the same cell. They remain distinct cellular cytoplasma. So they're distinct cells, but they're held together at the synapse. Okay? Now we can see a couple of curious things about the synapse here already by looking at this electron micrograph. The first thing is that we see, oh, there's a mitochondria at the synapse. Now, this is going to make sense because, as we'll talk about, a lot of the action that happens at the synapse is extraordinarily energy intensive. And so if you're going to have a really energy intensive thing happening at the synapse, you might as well bring your power plant closer. And so there's a mitochondria that we can see at the synapse because it's just live streaming ATP so that we can use them without having to transport them really far. This is also something that's very commonly seen in both the axons and also the dendrites of the cell. So like we said earlier, the cell body houses the nucleus stays at the cell body, it doesn't really move, but lots of other things are actually sent far away onto the far processes of the dendrites and the axons, and one of them is the mitochondria. There's power plants all over the neuron because it's so power hungry. The second thing that we can see is that the synapse, despite the diagrams that I'm going to be showing you for the next couple of minutes, is actually full of stuff. It is not at all empty, right? This is one of the really remarkable things about cells, if you think about it, because we draw all these diagrams, we have these dioramas, we have these cartoons, and it seems like there's a lot of space, there's a lot of water around in the cell, right? This is not the case in reality. In reality, the cell doesn't really want to waste space. And so the synapse, because it's such an active zone of activity, also has tons and tons of proteins. What are they? Well, we're going to start the process of talking through some of them and thinking about what they do, but this is an extraordinarily active area of research in neuroscience, people thinking about the various types of synapses and the different proteins that are active in them, what they do, and how they change in time. So, to review, we have a synapse, and we're going to be doing some terminology here just so we're kind of on the same page. When I use certain words, I want you to be comfortable using these particular words. The synapse refers to the entire structure where one neuron is connecting with another one. The one from which the communication is coming from is called the pre-synaptic cell, because that's where the information is flowing, right? So in this diagram, the information is flowing down. So there's a pre-synaptic neuron. And then on the receiving end, the other neuron is called the post-synaptic neuron. This terminology is specific to this particular synapse, because somewhere else in the brain, could even be, a couple of microns away, this yellow cell could be making a synapse onto the pink cell the other way around, right? And so in that case, for that synapse, the yellow cell might be the presynaptic neuron, and the pink neuron might be the postsynaptic neuron. So this terminology, to be clear, is specific to this particular synapse. Okay? One thing that is also something that is hard to see in cartoons sometimes is the synaptic cleft, the space between the presynaptic neuron and the postsynaptic neuron is extraordinarily thin, right? It's not some vat of water where things are kind of floating around and molecules are floating around. It's actually really thin, okay? And its spacing is actively maintained by a set of proteins for reasons that hopefully will become clear in a little bit. Now, one more piece of terminology here. In the presynaptic neuron, we have a set of vesicles. Vesicles is just a generic term to mean small membrane-bound compartments in the cell. Okay? In this particular case, we're going to see in the next couple of videos what it is that these vesicles do and how their function is so crucial and pivotal to the function of the synapse and how they mediate the transmission of electrical activity from the presynaptic cell to the activity in the postsynaptic cell. So preview is that the vesicles are actually full of chemicals, and so instead of having all electrical to electrical activity, we're actually going to have an intermediary step in the middle where there's chemical transmission, which is why this type of synapse is called the chemical synapse. Okay, so a little bit more on parts of the synapse, because I always really like knowing what sizes of things are, like I've tried to emphasize throughout this series of videos already. So it's just a couple of numbers to orient you in terms of how big, roughly speaking, things are. So a synapse, again in the mammalian central nervous system, tends to be hundreds of nanometers across. It can get bigger or smaller, okay? So there's definitely synapses that are much larger than this, and they can be smaller than this, but we're talking about the order hundreds of nanometers here. In comparison, the synaptic cleft is very thin, so we're talking about 20 nanometers, about a very small space that spans the space between the postsynaptic neuron and the presynaptic neuron. Each vesicle is going to be something like 35 nanometers, of which there are usually about hundreds of them that are ready to go in the presynaptic neuron. At any given moment, each of those vesicles might be holding, about 1,000 neurotransmitter molecules that are packed up and ready to go. Okay, so this is what the sizes of things are, and now we're ready to talk about in the next video how the synapse actually works as a dynamic structure. So to review, we have a mnemonic here, which is, I like to think about here in Seattle. We can look at a map of Seattle, and what you'll notice is that this is the map of Seattle where downtown Seattle is here. Okay, there is a body of water we call the Cut that goes from the Puget Sound saltwater to Lake Washington, which is freshwater. Here, the University of Washington, we're right here-ish in the University District, and there is a lake called Green Lake, which is in North Seattle. I like looking at this picture because I think it's actually a diagram of a synapse, right? So it's got the presynaptic terminal in the northern side, the postsynaptic terminal on the southern side, and there's even there's a little vesicle that's docked there. So if you have trouble remembering the synapse and the different parts of it, and you happen to know a little bit about the geography of Seattle, this is what the Seattle basically looks like, a synapse to me. Okay. So, key concepts that we have talked about that I will review very quickly to understand the neuron, the anatomy of the neuron, and the parts of the synapse. We talked about how neurons have multiple distinct compartments, and they have very different functional roles. The most relevant ones for us to remember are the dendrite, the cell body, the axon hillock, and the axon. And they're distinct both anatomically and also in terms of their function because they express different proteins. So the dendrites, generally speaking, are the inputs. They take in sensory inputs, and these are either from the external world, like photoreceptors in your eye or touch receptors in your hands, or they're inputs from other neurons, and there's lots and lots of them, right? So generically speaking, dendrites take inputs, and these electrical potentials propagate in a passive way without generating action potentials towards the cell body, also known as a soma. Now, when these action potentials are then generated as all or nothing events at the axon hillock, and once they're generated, if they're generated, they then propagate along the axon in the same way that we learned about in the previous video about axonal action potential propagation. And the axon is where all of the voltage-gated sodium and potassium channels are, which is what allows it to sustain action potentials. This is also where we would find myelination if the cell had any. And at the end of the axon, the cell communicates with other axons or other electrically active cells, for example, muscles, at structures that we call synapses. And we talked about the structure of those synapses. And so to be clear, the from neuron is always the presynaptic neuron, which talks to the postsynaptic neuron. And these terminology, these words are specific to that particular synapse. There's nothing intrinsic about a cell that makes it presynaptic or postsynaptic. Every neuron is talking to lots of other neurons. Neurons do not exist in isolation. And so they'll take turns being the presynaptic neuron or the postsynaptic neuron, depending on the structure that we're talking about. So we are all set now. We have the stage set, we have the terminology, we have the anatomy, and in the next video, we're going to get to the action of what happens at the synapse when the action potential arrives. What happens when it arrives? Tune in for the next video.